Hello, my name is Anne Jones and I'm the curator here at Creswell Crags. And in this video I'm going to be talking to you about paleo diet and reconstructing paleo diet. So essentially that is reconstructing what animals ate in the past. And we're mostly going to be focusing on mammals, but some of the techniques that we'll be speaking about are going to be applicable to other vertebrates as well. So I'm going to begin with showing you some of the items from our collections here and some of the techniques that can be used, and then we're going to head across to the British Geological Survey in Keworth in Nottinghamshire to speak to one of the scientists there about how they reconstruct past diets. So one of the techniques that we can use to reconstruct paleo diet is called dental microwear. So that is essentially looking at the tiny microscopic features imparted onto the enamel of the tooth when an individual was feeding. So that includes like scratches and pits, and it reflects the last few meals of an individual's life. So essentially what scientists can do, one method they can do is to take the tooth, clean it, and then they will use dental silicone, the same thing that dentists use. So it's this blue stuff inside here. Pop it onto the tooth, let that sit for a few minutes and then take it off. And then sort of the dental silicone will have the impression of the tooth. They then go into the laboratory and put some clear resin inside this mould here to get something like this. And then what they do is they look at it under a microscope with special lighting, and they count the number of features that they can see, such as scratches and pits and gouges on an area of the tooth. And that can help them to understand the past diets of an animal. So for example, here we have a spotted hyena, first lower molar, and a cast from the first lower molar as well. And if a hyena tooth has more pits on it, that might indicate that they were consuming more bone in their last few meals. So another way that we can reconstruct past diet of a species is through the damage that they imparted on bone. And the species I'm going to be focused on here is the spotted hyena. And they were really, really important during the Pleistocene in Britain because they denned in caves, they brought the remains of their prey into caves. So one way is we can literally look at what species were found alongside the hyenas, and it's likely that the hyenas were eating those species. But even more importantly is that they have the tendency today and in the past to consume bone. So they will crack bone, they will remove the ends of bones as well to get at the marrow inside. So we can look at the damage to the bones themselves. So we've got a classic example here. This is a long bone of a herbivore, and the edges of the bone are quite rounded and crenulated, so they're broken off the epiphyses, the ends of the long bone, to get at the marrow inside the shaft, so they've kind of started to hollow it out. So this is a classic example of gnawing by hyenas. And the teeth that they do that is their premolars, particularly their third premolar, because you can see here it's quite conical in shape, and this is an ideal shape that can help them to crack these really, really hard material of bone. And then finally I've got another hyena jaw here, and you can see that the bottom of the mandible, the jaw, has been broken away, and this also shows sort of the gnawing marks of the hyena. So not only were they consuming herbivores, but they were also consuming the bones of their own species as well. So we're going to stick with hyenas for the moment because I want to show you this. So this is a coprolite, which is fossilised poo of a hyena. So as I mentioned, hyenas consume lots of bone, and their digestive system is also very, very useful. So their digestive system can digest all but the inorganic portion of bone. So the calcium and phosphorus content comes out the other end of their poo, helps to make their poo this sort of whitish colour, and helps it to preserve for thousands of years. So scientists have extracted DNA from hyena coprolites, and there was a study by Bond and others who found some red deer DNA in a hyena coprolite, suggesting that the individual had been consuming some red deer. Hiya, so do you just want to maybe introduce yourself and what work you do here?
Sure, I'm Angela Lamb, and I'm a senior research scientist here at the British Geological Survey. And the work we do here is predominantly various isotopes that help us understand lots of different environmental and biological questions.
So we're here mainly today to talk about reconstructing past diets. So how do you use isotopes to reconstruct diets in the past?
So when, as a human or an animal, when you ingest food, you take on the chemical composition of that food, and the body metabolises that and stores it in the body. So your bones and your teeth and your hair and all your biological material will store that information, and then we can then look at that to get a picture of how your diet is translated in the body, and then we can build up a picture of who's eating who, and it will change the further up the food chain you get. And also there'll be changes in terms of how much fish is in the diet, how much meat's in the diet, whether you're eating a vegetarian diet. We can tell those different types of differences. And also you will be able to understand sometimes where an animal sits in the food chain and how that changes over time.
Fabulous. So in terms of the specific isotopes, which ones will you be measuring specifically for this purpose?
So we use 3 isotopes for this purpose: carbon, nitrogen and sulphur. So carbon tells us generally in terms of the environmental conditions, so that will change through warm periods and cold periods of ice ages. So, for example, the type of vegetation you'll get in an ice age, whether it's open grasslands or whether you have forested areas, you'll see a difference in the carbon isotopes in the diet. It will also be different whether you're eating seafood or terrestrial food. And then we use nitrogen as a sort of ecosystem structure isotope. So for every stage up the food chain you go, you will increase your nitrogen isotope composition. So someone eating, or an animal eating right at the top of the food chain, will have a very high nitrogen isotope composition, whereas the vegetarian diet will reflect just the lower values of the vegetation present. So those in combination can give you quite a good picture of ecosystem structure and diet. And we also use sulphur isotopes as well, and they again will give us some indication of whether you're eating seafood or not.
Fabulous. It's amazing, isn't it? Like how much you can understand of an animal that lived thousands of years in the past from just a tiny portion of bone.
Yeah, and especially now the technology has improved so much, we can work on much smaller material than we ever did before. So this is really helpful in terms of if we have very precious museum samples, we only need to take a very small amount of bone to give us those analyses.
So speaking of which, we've got a bone there from Dartmoor, I believe, in Creswell Crags, don't we?
Yes.
So in terms of sort of the sample you took from there, could you maybe explain a little bit about that, how it was done and where we took it?
Yes, so the first thing we do actually when we have a bone specimen is we can do a very sort of quick test just to check to see whether there's likely to be proteins preserved, because some of these bones obviously are hundreds of thousands of years old, so they're not always preserved, and we can just take a very small piece of the bone, we just scrape off a little bit of the surface to clean it, and then we take a drill and we use, this is a dental drill, so it has a very fine diamond tipped burr on it, so we can take very precise samples. I don't know if you can see there that this one has then been sampled because it passed the protein test, and we're taking a really very small amount of material away. Once we've done that, we take that into the laboratory and we then process it to extract the collagen. So we have to do a chemical process to really just remove the mineral part of the bone, leaving the protein, which we then freeze dry, and then that's ready to then process in the mass spectrometer.
So in terms of the mass spectrometer, how does that work? What exactly does that do?
So it's an instrument that measures really small differences in mass of different elements. So we have to get the samples into a form that can be introduced into a mass spectrometer. And we do that by combusting them, so we burn the sample in what's called an elemental analyzer, and then that cleans up the gas and it carries that gas into the mass spectrometer where those tiny differences are measured. And then those differences are compared to international standards, so then the data then is comparable across different labs and different organisations, so we can give the context of our samples in a much wider space.
So I think my final question then is, are there any sort of case studies or any research you've been working on recently that you're allowed to tell us a little bit about?
So we work on lots of different projects at the lab, and one of the projects that I'm currently working on involves the paleoecology of the wolf in Britain. So looking at how the wolf has evolved and how the structure of the wolf in those ecosystems has changed through different ice ages back through time in Britain. So this is looking at samples from Creswell Crags and lots of different museum samples to try and build a picture of how that has changed. So we are looking at the diets of wolves, but also of the other animals and fauna in the ecosystem that they were sharing the space with, just to see how that has built up and changed in different parts of the last ice ages.
Brilliant, so really watch this space for results.
Yeah,
then.
results are just coming in, so yeah, there'll be some exciting results hopefully soon.
Excellent, thank you very much.
You're welcome.
So what this video has shown is just a few methods that scientists can use to reconstruct the diets of vertebrates, particularly mammals, in the past. And there are many more methods and many more ways that scientists can begin to understand the lives of these individuals of these species. And I just find it so amazing just how much information we can get from just a few bones and teeth to understand how animals lived so many thousands of years ago.